Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Jennifer Lawson from Sartorius.
Thank you for the introduction, Leah. Hello everyone. Today I'm going to discuss the critical steps for potency assay development for protein therapeutics and how Sartorius supports this as part of our CHO platform for protein expression. So I'm going to start with protein characterization, then discuss potency, assay development and the bioassay life cycle, and then finish with a brief overview of the Sartorius CHO platform. So protein therapeutics, they need to be characterized, and the foundation for approval is really built on the robust analytical characterization, which proves the safety, the purity and the potency of the product. Developers need to demonstrate what is the protein, so to prove its identity. How does it work? Some molecules have relatively simple mode of action and some are very complex, but all modes need to be characterized. And how well does it work, so the potency. And this must be demonstrated on a batch-to-batch basis. The ICH-Q6B guidelines give developers guidance on what they need to demonstrate for their molecule, with the end goal being an in-depth understanding of the molecule's characteristics, which allow relevant specifications to be set that will then prove the future batch-to-batch acceptability for use. So a key part of the assessment is the potency of the drug, and simply put, this is the amount of drug that is needed to produce a certain response. And for biologics, we can assess this using a suitable bioassay. And when we talk about potency experimentally, we're generally referring to what's called relative potency. So this allows for the inherent variability within the biological system, so for example within our bioassay. So to account for this, we measure the potency against a standard, which is constant between the assay runs. So for this to be valid, the standard and the test material must be biologically similar, such that the test sample ultimately behaves like a dilution of the standard. And we have to meet several important statistical criteria in order to generate a valid relative potency result. However, that's not going to be the focus of the talk today. So how do we generate our suitable bioassay with which we can assess relative potency? So cell-based assays are complex, and before starting the development, we need to take several factors into account. So firstly, the why. Why do we need the assay in the first place? For potency assays, or is it to evaluate potential secondary modes of action? We've got a bit more freedom when the assay is purely for investigation purposes. What is the product? So what can we surmise about it from its biology? What else should we consider in terms of the sample type that we will expect and the matrix of what the sample will be in? And how will we develop the assay? So at this stage, Sartorius would identify a suitable system and propose a methodology. And it's important to consider the endpoint and the future need for regulatory compliance. We should make informed decisions about the technology that we're going to use and its suitability for future GXP applications, and also the complexity of the proposed assay system. At Sartorius, we have a wide portfolio of platform methods with a range of different endpoints to utilise, which speeds up this planning phase. Once the planning is completed, we would move to a proof of concept with the platform to identify what needs to be developed and optimised further. And then once that's completed, we confirm the finalised methodology and perform an initial assessment of accuracy. So during the bioassay development process, there are some key considerations aside from the general assay development considerations like cell seeding density and incubation times. that we need to think about when we're developing potency assays. And I've selected a few of these to discuss in more detail. And this is not an exhaustive list of everything that you need to consider, but it's a starting point for this discussion. So the first one is the mode of action. Our potency assay needs to correlate with the mechanism of action for the product and ideally be stability indicating. So we need to select a system that is functionally relevant. But we also need to balance this with the second critical point, which is the variability of the assay system. Bioassays, like we said, are inherently variable, so we're going to need our assay, but we need our assay to be robust and reproducible for what we're going to use it for. So an assay with a large number of steps or multiple primary cell types or complex biological reagents might be very physiological, but ultimately it's unlikely to be a good candidate for a GXP routine potency assay measurement. When we talk about cell-based bioassays, the cells introduce probably the arguably biggest source of variation. Our assay cells can be immortalized cell lines. They can be cell lines derived from tumours, ex vivo primary cells, or genetically engineered cell lines. Obviously, a cell line is really the preferred choice because this allows us to start building in some level of control over this source of variation. We could create master and working assay cell banks. We can standardise pre-assay culture conditions. It's also increasingly common to use thaw and use cells. So here you bank enough cells per vial for one assay run. And that even further controls the variation by standardising the passage number of the cells going into the assay. But continuous culture is also an option that can work very well. So I mentioned briefly on the previous slide about selecting the assay endpoint. And by endpoint, I'm referring to the readout to generate our result. So as an example, if we think about a common measurement in a bioassay, for example, proliferation of cells, there's a multitude of ways to measure proliferation, from simple dyes in a plate reader to more complex flow cytometry or even real-time cell analysis. And as mentioned already, we want ideally a very robust measurement, and we also want to choose a system, both hardware and software, that is suitable for GXP applications, so meaning it's got the capability to be compliant and to be validated. So the plate layout is another factor to consider. We can perform heat maps on our assay plate to look for bias by running the same sample in every well. And we should try to implement a design that prevents any bias through randomization. We do need to think about when designing the plate plan, we should bear in mind really how the assay will be performed. So will it be manually performed, or is it going to be using a robotic liquid handler system? And if the plates are going to be manually prepared, then we need to be able to strike a balance with enough randomization to prevent any position effects and what can be feasibly performed by hand on a routine basis. We don't want to introduce any more capacity for error by having a very, very complex randomization on our plate plan. So what about replicates? The more replicates we have, the more confidence we have in the results from a statistical perspective. But for that to be true, the replicates must be independent. So if we prepare one dilution from our test sample and we use that to fill, for example, 3 wells to act as 3 replicates, they're not truly independent from each other because they come from the same dilution. So if we want a real assessment of our sample variability, which is important when we look at our statistical analysis, we need to prepare each dilution individually. So for 3 replicates, we would have 3 independent dilutions from the test sample itself. And the difficulty with sort of pseudo replicates, which is the same dilution, can be that we could produce data that is artificially precise and therefore potentially fail our system suitability criteria for a sample or the standard, which would ultimately lead to a failed run. So in terms of the dilution series, as a general starting point, it's a good idea to have a fairly wide and consistent dilution series. with the goal to capture the upper and lower asymptote and our linear response. And once the other development conditions are set, we can then look at adjusting the dilution series to provide the best coverage. The statistical analysis and reportable value is obviously a critical consideration in assay development, but it can also quite often get ignored due to its complexity. As I said earlier, this webinar will not focus on this topic in any depth, other than to say that we need to consider the appropriate curve fitting model for our data. So generally the 4PL is a good place to start for bioassay data, but other models can be assessed for suitability depending on the data. Within our system suitability, we can set requirements for the goodness of fit, or the R-squared value. To generate an accurate relative potency measurement, our sample and our reference must also be parallel. And to test for parallelism, you can use the F-test, which is the preferred test in the European Pharmacopoeia, or equivalence testing, which is the approach preferred by the US Pharmacopoeia. And once we've satisfied our system suitability criteria, we can analyse the data from the run and produce our relative potency result. And we also need to define how we will generate our reportable value. Will this be from data combined from several assay plates over one run or multiple runs? Will our potency be calculated from each plate individually or from the entire data set across multiple plates? And there are a few specialist data analysis software programs that can really help here, but it is advisable to consult a bioassay statistician if you've got any doubts. So I'd like to share a couple of case study examples of potency assay development from the Sartorius labs. And these were performed as internal studies as part of our platform assay development, where we develop a platform method and then evaluate it with commercially available suitable drugs. And the first is the development of a potency ELISA. So with all the discussion previously on mode of action, why would we develop an ELISA and not a cell-based assay? Well, when you have a straightforward mode of action, so in this case, the blocking of the binding interaction between PD1 and PDL1 and PDL2, coupled with the need for a complex bioassay using primary cells to be able to demonstrate that in the cell-based system, a potency ELISA or a suitable binding assay could be used with your bioassay to demonstrate correlation of the results using this more robust assay. And we know that the potency testing for Opdivo, which is sort of nivolumab, the molecule we're used here, is based on three measures of the potency: a binding assay, a potency ELISA, and a cell-based bioassay. So how do we go about designing our potency ELISA? Well, the blocking mode of action is considered, and we chose to design our assay as a competition ELISA with PD1 to coat the plate, a constant concentration of biotinylated PDL1, and then a competing dilution series of our anti-PD1 molecule. So we can detect any bound PDL1 using streptavidin HRP. And at Sartorius, we make use of the excellent data analytics tools we have at our disposal in our MODA software to use a full factorial design of experiment approach, allowing a really efficient and thorough evaluation of the key parameters of the assay. And thinking about our key considerations earlier, you can see we're using a nice wide dilution series of our test sample at this stage to really try and capture the whole curve, and we've selected a robust endpoint on a GXP compliant system. So from our DOE, we can quickly identify the best parameters, and this allows us to move forward to optimize the dilution series before running an initial assessment of the accuracy of the assay. And this assay was then extensively qualified to determine the method performance. So in the second case study, we looked at development of a cell-based platform method to measure inhibition of proliferation using HER2 binding as our model system. So HER2 is expressed on some breast cancers, and targeting HER2 has led to some blockbuster therapies, so the monoclonal antibody Herceptin and the ADC Kadcyla. So for those of you who aren't familiar with this assay, it takes generally an extended incubation period, meaning it does need to be quite tightly controlled. So for this example, I'm just going to show a couple of key steps to give an idea of the development process that we went through. So firstly, we considered the cells, which cell type should we use? And we evaluated a couple of different cell types for expression using the Sartorius IQ screener. And we also looked at comparing a range of different cell seeding densities in the two plots shown here. We compared assay endpoints, two plate reader based examples, Alamar Blue and Cell Titer Glow were shown here. And we also looked at non-plate reader based readouts such as real-time cell analysis. And once we've assessed the different factors, we confirmed the method with both Kadcyla and Herceptin to demonstrate the broad-based suitability of our platform methods. And based on the preliminary work developed in the platform, we can then quickly optimize further for potential candidate molecules which target HER2. And we can also use the same systematic approach to apply for other molecules with similar proposed mechanism of action, but with a different target. So the life cycle of potency assay development, ideally it should be stepwise across the lifetime of development of the product. And the timeline here shows some key stages, but it's important to emphasize that product characterization should really be ongoing the whole time to ensure that we fully understand the molecule and the inherent variation between batches. And also to point out that the timeline here is for visualization purposes, so it's obviously not to scale. So some key stages are the system suitability studies, where we can start to understand the performance of the methods and set tentative system suitability criteria for what constitutes the test's validity on a day-to-day basis. And with this bank of data, we can also complete the determination of what we will use as the reportable value and finalise how our data will be combined from assay plates or assay runs. We should also early on assess critical materials. So critical materials are defined as materials which are a key component of the assay and are complex biologically derived molecules. So they possess basically lot-to-lot variation. Things like our cells, substrate, the antibodies perhaps that we might use within the assay. And these materials have inherent variability batch to batch, which can impact the ability of the method to deliver accurate and reproducible results. So we need to really assess that and understand how that variation impacts our assay. As developers move to phase 1, a GMP qualification is needed to determine the method performance capabilities for parameters such as the specificity, the linearity, accuracy and precision as required for the intended application. And this is sometimes also referred to as a phase 1 validation or an early validation. At Sartorius we use the term qualification. So a key point is really that a method can't really fail a qualification. It can and it should be re-optimized until it can achieve acceptable performance. Or we should reject it for the application and go back and start again. As the product moves into later phases, a validation is required and robustness assessments should form part of that. So performing the robustness assessment provides an indication of the reliability of an assay. So really we're assessing the capacity of the assay to remain unaffected by small but deliberate variations to the standard assay methodology. And you can also perform robustness earlier, prior to the phase 1 qualification if required. And the chosen parameters for robustness are based on knowledge of the assay and the associated risks, taking into account factors which might happen within the laboratory. At phase 3, we need to consider the GMP validation, and I think it's key to note that validation should never be a surprise. The protocol should have predetermined acceptance criteria to confirm the method performance capabilities, and the method acceptance criteria should be established before starting the validation. So your validation is not a discovery study. Acceptance criteria needs to be met by every validation run for the validation study to pass. And if a run fails a criteria, then we need to investigate that and look for an assignable cause for the failure. and to resolve that before the method can be considered fully validated. And the accepted guideline for the validation of assays has been ICHQ2, and this is the basis of the validation protocols which we use at Sartorius. So what types of assessments are included in a qualification or a validation study? Like I mentioned, we follow the ICHQ2 guidelines for validation as standard, although we can discuss with clients individually on their requirements if they have protocols that they prefer to use. And this example is actually for a very extensive qualification, but for a validation we would probably do something relatively similar, maybe just drop some of the concentrations and replace them with more 100% measurements. Because at validation, we're not really so much testing the range of the assay, we're confirming what we already know. So the assessments include looking at the accuracy, so how close the returned value is to the actual value. So if my sample has a relative potency of 50%, how close to 50% will my returned values be? How close are they to the centre of our target? The precision, which is the closeness of the returned values for multiple runs of the same test sample. So on our little target here on the slide, we can visualize precise data as all of our arrows are close together, but they're not necessarily on target. So precision should be considered on 3 levels. Repeatability, so comparing results generated under the same conditions over a short space of time. Intermediate precision, which is comparing within lab variations, so different analysts, different days, different equipment, etc. And reproducibility, which is the precision between different labs. We would also do 1 or maybe sometimes 2 specificity assessments. Assess the dilutional linearity, so that's the assay's ability to produce results which are proportional to the concentration of the sample. And the range, so what's the upper and lower concentrations of sample for which we can demonstrate the required accuracy, precision and linearity. And as we said before, a validated assay would also have the robustness assessment carried out as well prior to the validation study. The acceptance criteria are somewhat assay dependent, but by the time validation is reached, they should be quite well known. As a guide, a good bioassay will return accuracy of plus or minus 25% of the expected value and have intermediate precision within 20%. But realistically, we can see plus or minus 30% in some bioassays and percent CVs of 25. We'd also expect R squared of greater than or equal to 0.9, and that our bioassay could meet these criteria for a good range, perhaps 50 to 200%. So what is the Sartorius CHO platform, and how does this benefit drug developers? So we've combined the performance of the CELCA cell line development system with the fully integrated services for GMP cell banking and characterization, protein characterization and assay development, and our 4-cell fully optimized complementary media system to produce the CHO platform. So what does that look like in terms of the workflow? Well, here we can see the different stages, our CELCA cell line development, protein characterization, cell banking and biosafety testing. And really the point of the CHO platform is to guide customers to offer predictable results and to really accelerate the timeline. Why did we include potency assay development within this platform? Well, bioassays are consistently top of the list for outsourcing requirements, and that's really because they're complex to develop and also complex to run and complex to maintain. So our belief is that the sooner the development for this critical component is started, the better chance to avoid costly delays at later clinical phases. So we offer platform method solutions to reduce the development time by up to 50%. And we also have a specialised toolbox of methods for characterising immune checkpoint inhibitor development drugs. And alongside our assay development options, we are also very experienced at transferring in methods which have had anything from a very basic level of development upwards, so that we can then complete any optimization that's required, perform the qualification and validation studies. So to summarise, what is Sartorius offering with the CHO platform? We're an integrated provider. Our integrated analytics and assay development allows an early focus on these critical parts of the drug development process. Our platform allows seamless transition from cell line development to cell banking and characterization. It's designed to shorten timelines and save time on managing multiple suppliers, to reduce risk, and to offer cost reduction through bundling in comparison to individual services. And finally, we have dedicated client management to really help guide developers through the complex process. And with that, I'd like to thank you for listening and take any questions.
Okay, great. Thanks. So, first question is, what sort of endpoints can you recommend for potency assays?
Really there's no straightforward answer to that. It depends on the mode of action of the product. Like we said, the potency assay needs to demonstrate the mode of action, so the endpoint needs to be concomitant with that. A key factor is really to think about how robust the endpoint is for the future qualification and validation. And it's good to note that not all technologies is easy to validate as each other. Some systems are really more designed for discovery work and don't have the GXP capability that would be required. And the ease of data handling is also important, so you don't want to jump from multiple different systems. Data integrity and computer system validation are key focus areas for the regulators at the moment. So the systems that we use need to be appropriate for the task in hand. All in all, I'd say the endpoint should be considered based on the product and the purpose of the assay.
Okay. And how do you select the cell line for your assay?
We saw the cell lines quite often the biggest source of variability in our assay, so we shouldn't skip this assessment and just kind of pick one at random. I'd say there's a few key things to keep in mind when selecting the cell line. Again, the mode of action, how's the cell line going to demonstrate that? Does it express the right markers? Has it got the right internal machinery? And another thing to bear in mind is if you're using a cell line for example, ATCC or similar, then you need to consider the license that you need to be able to use that cell line for ultimately what will be a commercial purpose in lot release. So you would need a commercial license to be able to use that cell line for the future. And that can take a little bit of time to get that put in place, so it's worth to start that process early to make sure that you've got it in place for when you need it.
And what is the optimal number of replicates?
So this really depends on the assay itself and its level of variability. If you've got more variability, then more replicates would obviously help with that. As I mentioned, we should ensure that our replicates are true replicates and not pseudo replicates, so not the same dilution of the sample across multiple wells. True replicates will give a better understanding of the variability. They'll prevent issues with failing statistical analysis due to that being underestimated. So if you have very highly precise data, so for a concentration, then that very small amount of error can lead to failures in the F-test for the parallelism assessment, ultimately leading to a failed run. So I would say, in general, you don't want to be any less than 3 independent replicates, but potentially you would want more than that, depending on your assay.
And is it possible to perform these services with Sartorius independently of the cell line development platform?
Yes, absolutely. So we have these services available as standalone development services, inclusive of sample testing, qualification, validation. We can also offer stability studies. We have a full range of methods for protein characterization, which sort of complement the potency assay development. And we really see the benefit for customers to get it started as early as possible, which is one of the reasons why we include it with the CHO platform services to really, get customers to think about this at the cell line development stage at a good early part of the process. And to be honest, a good potency assay can be really an invaluable tool to assess the process development and the product quality throughout the development process. So there is a real benefit to having the assay in place early.
And in your slide on the potency, Eliza, you mentioned a quality control sample. Can you explain what that is?
Yep, sure. So the quality control sample mentioned in that particular assay is it's actually an independent preparation of the reference sample, which we run at 100% to act as an internal assay control. And then that allows us to set some system suitability criteria around about that, so to really check the validity of our assay on a day-to-day basis. Because we know what the expectation of our assay is on the accuracy that we should achieve for that sample. And that's actually, to be fair, one thing that I didn't discuss today, assay controls. These are very dependent on the assay type itself, but they're obviously critical to show the valid performance of the assay. And they form part of our system suitability criteria that we generate from our system suitability bank of data. So in general, having a second independent control sample is a good way to assess the performance of the bioassay on a day-to-day, run-to-run basis. And we should be trending the reference material once the assay is GMP. But a control sample provides added reassurance in the early and the later stages as well. So in this instance, as I said, it was actually an independent preparation of the reference standard, but it's also possible for you to use an independent sample with a known potency that you include on every assay as a control.
Okay, I think that may be related to this question. So, since not all pharmacopoeias give clear instructions, how do you usually set the preliminary and final assay acceptance criteria? What would be a good approach mathematically?
So I would say again, it's really dependent on your assay as to what your assay acceptance criteria is going to be, and it's dependent on your system suitability data. There's no point setting acceptance criteria that your assay can't achieve. That would show that your assay is not good enough, and you should go back to the drawing board. So really, what you want to do is look at your system suitability data and take your acceptance criteria from there. And anything more than that, I think you're really starting to get into, the statistical analysis of your data. And for that, I think it's a good idea to talk to a biostatistician if you've got questions on how to do that.
Okay, well, I think we have time for just one more question. If we didn't get to your question, we will be passing all the questions on, and Jennifer can follow up with you directly. The last question for the webcast is, what validation criteria on repeatability and precision do you propose?
So, what validation criteria on repeatability and precision do we propose? So again, it's quite often dependent on your assay, but in terms of our intermediate precision, then you want to look at the percent CV. So like I said, for the bioassay, a good bioassay would be within 20% of your intermediate precision between your replicates. But like I said, bioassays are complex. Sometimes the best that you can achieve is 25%. And I think that, if you've got a good justification for that, then that is okay to go above the sort of what is considered good of 20%. 20% is easier to achieve when you've got an assay system that's very well controlled, or you maybe aren't using cells within that assay, or you're using thaw and use cells or something like that. So that, I would say, is a good standard for your precision.
Okay, well, thank you, Jennifer.
Thank you.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.